Hello students, I am C. Pavani, Assistant Professor at B.V. Raju Institute of Technology from Pharmaceutical Engineering Department. In today's video, we are going to learn about metabolism of fatty acids and most important metabolism, beta oxidation of fatty acids. Beta oxidation of fatty acid is a key metabolic process that breaks down fatty acids and produces ATP. This ATP is used as energy for other metabolic processes. And this beta oxidation is a catabolic process. Catabolic process is a process which converts heavier or long chain fatty acids into shorter molecules, or in this process we get 2 carbon chain acetyl coenzyme A as end product. And I missed this process, this beta oxidation occurs in cytosol of prokaryotes and mitochondria of eukaryotes. And acetyl coenzyme A, that is the end product of this beta oxidation, is utilized in the Krebs cycle, and it is also called as citric acid cycle. And along with the acetyl coenzyme A, this beta oxidation process also produces NADH and FADH2. NADH is nicotinamide adenine dihydrophosphate, and FAD is flavonine adenine dihydrogenase. These 2 coenzymes are utilized in majority of electron transport chains. This process is referred as beta oxidation because generally fatty acid contains a long chain hydrocarbon chain. Along with that, it contains carboxylic group. Carboxylic group is considered as functional group, and the carbon which is attached to the functional group is termed as alpha carbon, and carbon which is attached to next to alpha carbon is termed as beta carbon. And the oxidation occurs at the beta carbon of the fatty acid, hence it is termed as beta oxidation of fatty acids. As I already mentioned in the previous slide, it occurs in the mitochondria of eukaryotic cells, and it is crucial in maintaining energy balance, especially during fasting and exercise conditions. And the majority of molecules that are made in the body are formed that stores energy in the form of fats. And these fatty acids are typically produced in the liver of human body. Along with that, they are also taken through dietary sources of food. We have in the fatty acid, there is 1 or more double bonds which are termed as unsaturated fatty acids. If there is no double bonds, then it is termed as saturated fatty acids. And these fatty acids are stored in the adipose tissue in the form of triglycerides. Triglyceride is nothing but one glycerol molecule . Triglyceride is nothing but one glycerol molecule is attached to the 3 fatty acid long chains. And during this beta oxidation processes, this triglyceride enters into a cell by converting into 3 free fatty acids. So the triglycerides that are converted into free fatty acids enter into the blood. From there, wherever the energy demand is arisen, there it enters, and there it undergoes beta oxidation to produce energy. The mechanism of beta oxidation of fatty acid. Beta oxidation has several stages. There are 3 important stages. First stage is activation and transport of fatty acids, and second stage is beta oxidation cycle, and third stage is generation of ATP and metabolic intermediates which are utilized further for the energy. This activation and transport of fatty acids occurs inside the cell. This occurs inside the cytoplasm of the cell. Beta oxidation cycle occurs in the mitochondria of the cell. So, the first step, it has to activate and enter from if you consider this as cell and this is cytoplasm, and you have a mitochondria inside the cell. So after entering into the cell, in the cytoplasm, it has to get activated. After activating, it will enter into the outer membrane of the mitochondria. From outer membrane of the mitochondria, it will enter into inside the mitochondrial membrane. There, this beta oxidation process occurs. Here you can see the beta oxidation process. Let's start with the stage 1. So, as we discussed, the fatty acid from the blood circulation, it has to enter into the cell. So before entering into the cell, fatty acid is not a free fatty acid. It is available in the form of triacyl glycerol or triglyceride. It is also called as triglyceride. This triglyceride has to break down into glycerol and 3 free fatty acids. Why? Because glycerol is a heavy molecule. It cannot percolate through the cell membrane. So when it can break into glycerol and 3 different fatty acids, then only the fatty acid can enter into blood circulation. After entering into the blood circulation, it will enter into the specific cell where the metabolism occurs. After entering into the cell, it has to enter into the mitochondria. So, from entering from cell, from outside the cell to inside the cell it's easy step. It can enter easily. But here, from cytoplasm of the cell to mitochondria, it need to undergo activation step. Activation is nothing but fatty acid is converted into fatty acyl coenzyme A, where we term this as activation. This step is termed as activation step. After converting into acyl coenzyme A, only it can enter into the mitochondria of the cell. So, acyl coenzyme A is the only form that can enter into the mitochondria. And from mitochondria, you can see here, from the cytosol of the cell to enter into inside the mitochondria membrane, we call it as matrix. We have one more barrier. This is outside the mitochondrial membrane. We have one more barrier. Here acyl coenzyme A have to bind to the carnitine to enter from here to here. If it won't bind to the carnitine, it can enter only up to the outer membrane of the mitochondria, but it cannot go to the inside the cell. So from cytosol to, it can enter only to the outside of the mitochondrial membrane, but it cannot go to the inside of the cell. To enter into inside of the cell, it has to bind with the carnitine, which is present outside the membrane. And after binding with the carnitine, with the help of carnitine palmitoyl transferase 1, it will enter into the outer mitochondrial membrane. After entering into the outer mitochondrial membrane, acyl carnitine is converted, is bind to the carnitine palmitoyl transferase 2. After binding with the help of this CPT2, it will enter inside the mitochondrial membrane. Okay, so this process is possible only when it can bind with the carnitine. After entering into the inside the mitochondrial membrane, again it has to convert into acyl coenzyme A, because acyl coenzyme A is the only molecule which can metabolize and convert it into acetyl coenzyme A. So again inside the mitochondrial membrane, it gets converted to acyl coenzyme A. And as I mentioned, this inside membrane, it stops the entry of acyl coenzyme A, and we call this as carnitine shuttle. So carnitine shuttle is nothing but blocking entry of acetyl coenzyme A into the mitochondrial matrix. Coming to the stage 2. First stage is activation and transportation of acetyl coenzyme A into the mitochondria. Second stage, once the acetyl coenzyme A is entered into the mitochondrial site or matrix, then it starts converting into acetyl coenzyme A through 4 steps. First step is oxidation, second step is hydration, third step dehydrogenation, and fourth step is thiolytic cleavage. So let's start with the first step. Here you have the initial compound, that is fatty acyl coenzyme A. This fatty is replaced with 4 other terms depending on the length of chain, length of carbon chain. It is mentioned as 4 other terms. Let me write that for you. So the initial compound is fatty acyl coenzyme A. Depending on the length of carbon chain, this fatty is replaced by the terms lauric acyl coenzyme A if the number of carbons are 12, and myristic acyl coenzyme A if the number of carbons are 14, and palmitic acyl coenzyme A. Depending on the number of carbons, it can be lauric, myristic, palmitic, stearic, or arachidic acyl coenzyme A. Let us consider just briefly, I will give the outline. Fatty acyl coenzyme A, in the first step it undergoes dehydrogenation. You can also call it as oxidation or dehydrogenation. So this is the first step where it undergoes dehydrogenation, removal of hydrogens at the beta position and alpha position as well. So 2 hydrogens are removed at alpha and beta positions, and you will get a double bond in the first product. This process occurs in the presence of acyl coenzyme A dehydrogenase. This is the enzyme which catalyzes this reaction, and here the removed hydrogens are taken up by FAD and it gets converted into FADH2. Again, in the second step, we have hydration process. Hydration is addition of water, where this trans delta square enoyl coenzyme A is converted into beta hydroxyacyl coenzyme A, and here the water is attached at the beta position. Hydroxyl group is attached at the beta position in the presence of enzyme enoyl coenzyme A hydrotase. And in the third step, we call it as dehydrogenation step. Again, dehydrogenation is removal of hydrogen molecules. The hydrogen molecules are removed from the beta carbon. Again, this hydrogen is taken up by the NAD plus and it gets converted into NADH and H plus. This process occurs in the presence of enzyme L-hydroxyacyl coenzyme A. This process occurs in the presence of enzyme L-hydroxyacyl coenzyme A dehydrogenase. After that, beta keto acyl coenzyme A, we call it as beta keto because you can see there is a formation of ketone group at the beta position. Here there is a formation of hydroxyl group at beta position. So this beta keto acyl gets converted into fatty acyl coenzyme A, and also it releases acetyl coenzyme A, which is the main required end product of this beta oxidation procedure. So this happens in the presence of thiolase. Thiolase, we call it as cleavage. Cleavage is nothing but breakdown of molecule into 2 different molecules. So, and also along with this, along with thiolase, it also requires coenzyme A, thiocoenzyme A. In the presence of thiocoenzyme A and thiolase, it gets converted into acetyl coenzyme A, the end product. Okay, this acetyl coenzyme A is used in the Krebs cycle for the further energy production. And the remaining molecules are again converted. If the fatty acid is 16 carbon here, then you will call it as palmitic acyl coenzyme A, and after this whole process, here you will get a 14 carbon fatty acid and 2 carbon acyl coenzyme A. Okay, 14 carbon fatty acid. This 14 carbon fatty acid will again undergo the same cycle for the second time, and it gets converted into 12 carbon fatty acid and 2 carbon acyl coenzyme A is produced again. Again it co-same, so 10 carbon, so on, up to it gets converted into 2 carbon acyl coenzyme A, 2 end products at the end. In stage 3, all the produced molecules. So what are the end products of beta oxidation? We have acetyl coenzyme A molecule generated as end product from each cycle, and this acetyl coenzyme A is utilized in the Krebs cycle. We also call it as TCA cycle as well as citric acid cycle, that is further converted into energy through ATP. Next, the remaining NADH and FADH which are produced through this beta oxidation process are utilized in the energy electron transport mechanisms. And all this, like each NADH is equivalent to 3 ATPs and each FAD is equivalent to 2 ATPs. So if you see the overview, in the adipose tissue, we have triglycerides. The excess energy is stored in the form of triglycerides. Those triglycerides are converted into 1 glycerol molecule and 3 fatty acid molecules. Each fatty acid will enter into the cells. It gets converted into, or it gets activated into fatty acyl coenzyme A in the presence of thioesterification process. And after converting in the presence of coenzyme A as well as thioesterification process, after converting into fatty acyl coenzyme A, it tries to enter into, this is cytosol of the cell membrane, cytosol of the cell. From cytosol of the cell, it will try to enter into outer mitochondrial membrane, and from there it will enter into inner mitochondrial membrane. So where beta oxidation cycle takes place, it takes place in the inner mitochondrial membrane. And in the outer mitochondrial membrane, it gets transported from cytosol to inner mitochondrial membrane with the help of carnitine. And the process of blocking entry of acetyl coenzyme A from cytosol to inner mitochondrial matrix is termed as carnitine shuttle. Okay. After entering into inner mitochondrial membrane, the whole beta oxidation procedures occurs with the 4 steps. One is oxidation, hydration, oxidation, and thiolysis, cleavage. This oxidation is also termed as dehydrogenation. So oxidation is also termed as dehydrogenation even here. So after all this process, it gets converted into acetyl coenzyme A, which is the main required end product of the beta oxidation. So what is the first product of beta oxidation? Fatty acyl coenzyme A, and this fatty acid chain depends whether it can be lauric, palmitic, myristic. If whatever the may be the carbon chain, it has to get converted into 2 carbon acetyl coenzyme A as the end product. This is the end product of beta oxidation. So until it completely gets converted, this 14 carbons into 2 carbon acetyl coenzyme A, the cycle goes on. It is a repeated cycle. So coming to significance, this beta oxidation process is very important because this the metabolites produced in this metabolism is used for the generation of energy. It is used in the Krebs cycle. It is used in the electron chain transport of many cells. So it is utilized in the production of energy and maintenance of energy balance during heavy processes of the body. And the glucose levels, whenever the glucose levels are low in the body, the fatty acid get converted into the acetyl coenzyme A. And this acetyl coenzyme A is used in the citric acid cycle, and which can be create reducing equivalent NADH as well as FADH. NADH and FADH and as well as ATP. All these 3 are utilized in the electron transport chain. So the key points are, today we discussed what is the importance of beta oxidation metabolism, why we call it as beta oxidation metabolism, as the oxidation occurs as beta carbon of the fatty acid, we call it as beta oxidation. And second, we discussed how the fatty acid is transported from adipose tissue to inner mitochondrial membrane, and we discussed beta oxidation steps inside the mitochondrial matrix. And at last we discuss significance of this beta oxidation. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.